So am I the last person before drinks? Oh, you are. Haha. All right. Well, good afternoon. I know it's been a long day. It's been interesting. Some of you, I've actually met a number of you. There's some really bright lights, so I can't recognize you. And as many of you know by now, I'm very new to this business. I spent most of my career in the small molecule space. And about 25 years ago, I started to be interested in stem cells or cell therapy. And I thought back then it was a bit early. And I will tell you my impression of the last, certainly the last day or two, is that it would seem to me that stem cells certainly have come of age. Can I have the first slide, please? So what is StemBioSys? So StemBioSys is a company that I got involved in this year. The company is headquartered in San Antonio, Texas. And when I first heard about the company, I wasn't quite sure what to expect. And as I began to dig into it, this was about this time last year, I discovered that the company's principal mission is to isolate and expand and deliver stem cells. And as it turns out, when I first began to look at the science, I am not a cell biologist. I thought, well, I best take this technology, and I dragged it off to MIT and met with a guy who many of you know named Bob Langer and Dan Anderson at MIT, and we kind of crawled through the fundamental science to figure out what was there. And we concluded that, in fact, the company had come up with some platform technology that had tremendous potential. And that led me to get involved with StemBioSys. Could I have the next slide, please? Is there a button here I'm supposed to push? Anyway. So what is it that the company has from a technology point of view? So there are basically two platforms, as I see it. So the first is a technology platform that is what would be called an extracellular matrix. What do I do? This thing? Okay. All right. So in any event, so we have a matrix, and the matrix is basically the fundamental element of the company's platform that allows us to isolate, expand, and maybe one day deliver stem cells. And the way I see it is it falls into two categories. There's the extracellular matrix, and then we have a tissue-specific matrix. And so these two platform technologies are the technologies that we're developing now internally and that we intend to take forward and use those to isolate and expand stem cells. And then obviously we have a bigger vision in mind. We do intend to migrate into developing our own therapeutic stem cells, both independently and interdependently with partners. And so we've also identified a rare stem cell from human umbilical cord blood. Oh, I'm not doing something right. Do you want to advance the slide, please? So this is a video, and it's often said sometimes that pictures will say 1,000 words. So now it's going. And on the left, what you see is traditional 2D technology, and on the right, what you see is StemBioSys' platform technology. And as you can see, the cells on the right are moving. They're moving rapidly. And so and maybe, do you want to replay that one more time? There it goes. All right. And so the conditions are identical. This is all filmed simultaneously. So again, what you're seeing are the same cell populations. What you're seeing is performance on conventional technology, and this is the performance on StemBioSys' extracellular matrix platform. Next slide, please. So the way we characterize the platform is it's a three-dimensional matrix. And it's deposited by marrow cells, and what we're trying to do is create an environment that's identical to the environment that actually has that causes these stem cells to become basically to be in a normal environment. And the technology obviously has also been what's allowed us to isolate our stem cell from cord blood. So this platform technology today is in development. We are looking to take it a bit further, and so, can I have the next slide, please? To develop it into a platform that can be then used to produce specific tissue. And again, this is all very early stage, and what I'd like to do is maybe take you a bit further as to what our mission is here, why we're at this meeting. Could I have the next slide? And as I said at the outset, we have identified a stem cell line from cord blood that we'd like to develop into for therapeutic indications. So where exactly are we? So where we are today is we've created a matrix. The matrix is, at this point in time, a phenomenon that we've observed. We've patented it. We've begun to build an intellectual property estate around it. We've begun to understand it. We're working with MIT and with UT, San Antonio, to develop a better understanding of how exactly does this work, how much of this is related to the proteins that are certainly part of the matrix, and how much of it relates to the structure, and then how much of it is all the interrelationship between the proteins and the structure itself. And so that process is underway, and that actually has just begun. There's a fair bit of work to do, but in the long term, the goal is to come up with a biosynthetic or a product that can be made reproducibly, cost-effectively, consistently, and on scale. And so that project is very fundamental to the business. Could I have the next slide, please? All of this takes capital, and obviously capital is required. We're all looking for capital. How do we find the money we need to go forward? And so that typically comes down to people. So if our core technology platform would be our rare stem cell coupled with our platform that we feel has unique properties, then it comes down to the talent that we have in the boardroom. And the talent we have, our co-founders are Steve Davis and Xiaodong Chen. Steve's actually here and would be delighted to talk to you. And Xiaodong Chen. Xiaodong is the stem cell scientist at UT who discovered this phenomenon, developed the original patent position, and is still working with us to this day. Steve was his co-founder, and Steve provided actually the business sense that the company had early on, and that helped the company attract its initial capital. As I mentioned at the outset, Bob Langer has joined the board of directors, and he chairs the Science and Technology Committee of the board. And John Fike with DPT, Gary Frazier was the CEO who turned around OSI Pharmaceuticals. Paul Costella is the managing partner for Targeted Technology Fund, and that Targeted Technology Fund is our lead investor. Like many of you, we're in the middle of a Series A financing that we've decided to upsize. There's a little bit more demand than we thought, and so we're pleased with the way our financing is going. We have a woman named Jean Cheever who was previously with Bristol Myers Squibb, but she represents the Series A investors, and then me as the CEO. Next slide, please. The key officers in the company, certainly Cy Griffey. Cy is here, so if you'd like to talk to Cy, you're welcome to do that. He's our Chief Operating Officer, as I said. He's got considerable experience in certainly cell technologies. And Mark Horsey comes to us from Genzyme, previously from Dell Computers, but we're more interested in the Genzyme experience. Next slide, please. And we have a few advisors. So the Kathy Berzik, you may know, was with KCI for many years and is currently a director of Becton Dickinson. Prior to that was with J&J. And the others, as you can see, the last person on the list is a guy named Bob Hutchins. He's here as well. And we're beginning to learn more and more about the business and who's doing what in this market. And he's a retired partner with Booz & Company out of New York. Next slide, please. And so if I were to circle back and what is it we're all about? So we're a platform company. We believe we have a cell environment that we think has unique properties. It appears to have unique properties with respect to isolating, expanding, and delivering stem cells for therapeutic applications. We have a rare stem cell that we think has potential as a multipotent stem cell. We have some very talented people in the company and surrounding the company, both in the boardroom and in the advisory board. And we have access to capital, and we're looking to expand relationships in the community. Any questions? There was one earlier. Somebody asked me, who I actually had bumped into before, about my relationship with the venture community. So yes, I think it was on one of the slides. I am a partner in SV Life Science Funds 3, 4, and 5, and I am an associate partner in Targeted Technology Funds. Yes?
I have a quick question.
Yep.
So, can you disclose what your extracellular matrix proteins are? What comprises your matrix?
Well, I can't. The reason I can't is that I don't know that we're 100% sure, but that's a fine question. So at the outset, what I was saying was the phenomenon is very clear. So it's been well tested. So we see what we see. I think to your point, the reason that we've engaged, we're doing a partnership between MIT and UT is we're trying to understand what in that protein collection is responsible for what's going on and how much of it is the proteins versus the geometry. And it turns out, I thought it was a fairly simple analysis, turns out to be very complicated. But it's also, we think, an opportunity for intellectual property, not only for us, but also for people we partner with. We think there's some interesting elements to that. Make sense? Okay. Any other questions? Well, thank you very much. As I said, Steve Davis is here and Cy Griffey is here, Bob, and we'd be delighted to talk to you.